And I'm already in progress. I'm going to go ahead and introduce our speaker today, Abby. Let me see here. Abby Gehring is a grad student at Florida Tech studying chemical oceanography in the Fox Biogeochemistry lab. She is originally from Illinois and moved to the area for school at FIT in 2016. Since then, she has developed a love for local environments like the Indian River Lagoon and has been a longtime advocate for environmental conservation. She is working on pursuing her master's with a career goal of studying water quality. And what we're going to learn about today is her master's thesis that is incorporating the bottle cap project, which many of you have heard about, and how that is going to impact nitrogen levels in the lagoon. So without further ado, Abby, go ahead and take it away.
Hello, let me share my screen really quick.
Perfect.
Can you enable participant sharing?
Oh, 1 second. Let me see. All right, give that a whirl.
Awesome. Can you
Perfect. Okay.
See the
Sorry
screen?
for the technical glitches, guys.
No worries. Are you able to see the screen all right?
Perfect.
Awesome. Well, thank you so much for the introduction and for allowing me to come talk to everyone today about our bottle cap project that we're using to restore natural nitrogen removal in the Indian River Lagoon. As you mentioned, I'm a graduate student and I work with my advisor, Dr. Austin Fox. So if you've heard about this project, I'm really excited to kind of give you some updates and tell you what we've been working on. And if you haven't, I'm just as excited to let you know about it, tell you some of our motivation and kind of where we're planning on taking this. And so we'll just get started right away. As I'm sure many of you know, eutrophication is a problem in the Indian River Lagoon, as well as in coastal ecosystems around the world. And so when I say eutrophication, this is just a fancy way to describe an excess in nutrients. So too much nitrogen, too much phosphorus. And so this has many different implications of its own. We have an increase in nitrogen and phosphorus to a system. This can fuel something like an algae bloom. And with an algae bloom, there's many different negative consequences it can have. So for one, you can see that obviously there's not much water clarity, and so sunlight is not able to reach your submerged aquatic vegetation like seagrasses, and they won't be able to photosynthesize and produce oxygen. Eventually, these algae blooms will die and decompose, and when this happens, the bacteria work to decompose this material, use oxygen in the water column, and this can lower your dissolved oxygen concentrations. And so in severe cases, you might reach hypoxic levels, and this just means dissolved oxygen levels below 2 milligrams per liter, or in extreme cases, you can have anoxic events where there's no oxygen. In these cases, you might start to see something like a fish kill. And so unfortunately, the lagoon has had its share of algae blooms. These images in the background were taken up near Cocoa Beach and Merritt Island towards the end of last year. So thank you to my colleague Connor Wong at Florida Tech for giving me these two pictures on the right. And you can see that in this instance, we were seeing some minor fish kills up in Merritt Island. And overall, this increases the organic matter in the system, and this can have different impacts on the biogeochemical processes, and it also impacts our sediments as well. So as I'm sure many of you are familiar with Indian River Lagoon muck. And so this sediment has accumulated throughout the lagoon. It's very organic, rich, fine-grained sediment. And we locally call it muck. Some different areas might refer to it as more muddy sediments. But this is what it looks like in this picture up here. And let me just tell you, you should be very happy that you cannot smell this picture. And so it's very dark color, there's not a lot of oxygen going in, and it creates these hostile environments within the sediment on the lagoon floor that's not favorable for natural processes that are supposed to be taking place. And so our ultimate goal is to understand these natural processes so we can work towards promoting restoration efforts that can further enhance natural ecosystem services. And so when I say natural processes in this context, I'm going to be referring to biological processes known as denitrification, nitrification, and anammox. And so bear with me. I'm going to walk you through these processes and keep it as simple as I can. So with nitrification, this is an anaerobic process. So it requires, or this is an aerobic process, it requires oxygen. And you have two different groups of bacteria involved. It's a two-step process. And the first group of bacteria, Nitrosomonas, they're able to convert ammonia or ammonium, which is one form of nitrogen, to nitrite, which is another form of nitrogen. And from here, a different group of bacteria known as Nitrobacter are able to convert this nitrite to nitrate. And from here, you have heterotrophic denitrifying bacteria, and they're able to take this nitrate and reduce it to nitrite and subsequently to N2 gas. And so it's important, it's part of the natural nitrogen cycle, and when these different forms of nitrogen are converted to N2 gas, it's able to diffuse through the water column back into the atmosphere, which is already 78% nitrogen. There is another process known as anaerobic ammonium oxidation, or ANAMOX. This is known to be a more energy efficient process because it happens in anoxic conditions, so when there's no oxygen, and these ANAMOX bacteria are able to oxidize ammonium directly to this inert N2 gas using nitrite as the electron scepter rather than oxygen. And so might be a little complicated. Overall, though, big picture is that these are natural biological processes that remove nitrogen from the system. They're part of the natural nitrogen cycle. And they do occur in coastal ecosystems. And so these different processes are responsible, and they have the potential to be responsible for removing a large fraction of external nitrogen loading. Now, it's important to remember that these processes occur mostly in the sediments at the oxic-anoxic interface. And so when I say the anoxic-anoxic interface, this image is kind of giving you a picture of what I'm talking about. So if you're looking at the sediments from a side view going down, in healthy sandy sediments, there's usually a layer of oxygen within these sediments. And then you keep going a few centimeters further down into the sediments, and you start to lose that oxygen, and you get your more anoxic regions. But that middle ground right there, that interface, is really important for a lot of biogeochemical processes that take place. And this is why it's so important to promote healthy sediments, because it's ultimately going to impact the overall water quality. And so there's a lot of arrows and things going on in this image. Don't want you to get overwhelmed, but the main three processes that I talked about is nitrification, which you can see it's happening in that oxic layer. I told you that it requires oxygen, so it makes sense that it would happen up in this top layer. And that provides nitrate for the other processes like denitrification, which is signified by the aquamarine arrows, and ANAMOX, which is shown in this orange with the orange arrows. And so big question is, if these are naturally occurring processes, then why aren't we seeing more natural nitrogen removal in estuaries? Well, for starters, estuaries have a lot of variable environmental conditions. And so we have temperature gradients, salinity gradients, we have multiple different sources of freshwater input. And so all of these sediment biogeochemical processes are strongly impacted by these different parameters like temperature, oxygen, salinity, and pH. And so an example of how variable it is, this is actually data from one of our networks, our sensor networks in our lab that we've been working to develop. And we're using dissolved oxygen sensors to monitor bottom water dissolved oxygen. And so St. John's has sensors located near the surface water, but we're looking at the water that's kind of directly above the sediments along the bottom. And so this data is collected from two of our sensors, one deployed directly above sandy sediments, which is indicated by the green line, while the other is above muck sediments or mud sediments. And that dashed line down there, that is signifying the hypoxic levels, so less than 2 milligrams per liter. And so you know there's a lot of fluctuation in this graph, and that's normal. It's normal to see day and night changes. So these are called diurnal changes. And if you think about it, it makes sense because during the day you have sunlight that drives photosynthesis and you have oxygen being produced within the water column. But at night, there's no sunlight, there's no longer photosynthesis taking place, but you have organisms that are continuing to respire and take up oxygen in the water column. And that's when you start to see these decreases. Now, in some cases, you might even with sandy sediments, you could see hypoxic, or even in some cases, if you have something like an algae bloom, anoxic conditions. But you can see in this graph that much more frequently these muck, the water right above the muck is going hypoxic and anoxic quite often. And it's also known that salt can inhibit these processes. It inhibits nitrifying bacteria, and so this further inhibits the other processes that might follow, like denitrification or anammox. We've also degraded the habitats that these processes occur within, and so I've mentioned eutrophication and this increased organic matter loading. This can decrease the oxygen concentrations within the water, which will inhibit nitrification, because I did say that it requires oxygen for this process. And overall, hypoxic or anoxic events can decrease the efficiency and the role that these processes like nitrification and denitrification can have as a sink for nitrogen. And so sediments are supposed to be a sink for nitrogen. Naturally, that's how it's supposed to be, rather than a source. But in some cases, we'll talk about this later with the muck, they can actually become a source of nutrients. The muck has lower oxygen availability, but it also has higher sulfide concentrations, and these are also known to inhibit the bacteria that carry out these processes. And so if you were to take a dissolved oxygen probe and you were to slowly place it into healthy sandy sediments, you're likely going to see within the first couple millimeters or centimeters, there is oxygen. And as you move further down, that oxygen gets depleted. However, if you were to take this probe and you place it into muck, there is automatically no oxygen available. And so we've lost that oxic surface layer in these sediments that drive a lot of these biogeochemical processes. And so getting a little bit into more of the research that we do in the Fox Biogeochemistry lab at Florida Tech, we do a lot of work with water quality, and my advisor, Dr. Austin Fox, specializes in nutrient fluxes and trace metals. And so we do a lot of work with these muck sediments. Specifically, we work to probe and identify some of the major muck deposits in the lagoon. And this is one of a picture from recently. We were out in the field. That is my advisor, Dr. Austin Fox. And you can see we have our probing pole. It's as high as 6 meters. And in this case, the thickness of the muck was taller than he is. So it was around 3 meters in this spot. And once again, just be happy that you cannot smell this picture. And so what you're smelling when you smell muck is that sulfide concentrations, and we said that that can inhibit these processes. When we're out here probing for muck, we'll also take sediment samples to bring back to the lab and analyze to see how much organic matter is in it, the water content, how much calcium carbonate, and we'll also take samples to extract the water from the muck so we can look at what nutrient levels are like within these sediments. We do monitor nutrient fluxes using things like benthic chambers, and so we can see what's going on in sandy sediments versus muck sediments. And through this, we've learned that muck sediments are a source of nutrients, and virtually all of the nitrogen that is expelled from these muck deposits is in the form of ammonium. And this happens to be the form of nitrogen that's preferred and promotes species like Oriumbra. We've also found that in low oxygen conditions, the muck again becomes a source of phosphorus. And so we're able to learn a lot about these, and all of these different processes and what we're learning going on in the muck helps us understand how we can further promote ecosystem services. As I mentioned before, we are building this network of sensors in our lab to monitor bottom water dissolved oxygen concentrations throughout the lagoon. So this is just a map showing you where our current sensors are at. And this project actually stemmed out of a project working to understand the impacts of aeration, to see if directly adding dissolved oxygen to the water column can improve the surrounding water quality. And in short, the study, yes, we found that it does improve water quality to an extent in the surrounding areas. However, we were approached and asked if we can somehow figure out how to make these processes better. So if oxygen can promote these processes, is there a way that we can actually enhance them more? And so that's how we kind of got into this realm of nutrient treatment. But overall, all of these things that we're studying in the lab come together, and they help us understand these natural estuarine processes and what's happening in the sediments and what's happening in the water column, because overall this is going to impact your water quality. So back into this project, and so I've mentioned that these are natural biological processes, so they are known about, they are established, and they are even used by treatment systems like wastewater treatment facilities, recirculating aquaculture systems, and large marine aquarias. And so unfortunately, we have yet to find really a treatment system developed that helps treat natural water. And so there's a lot of difficulties, like I mentioned, with the variable conditions, with salt complicating and inhibiting the processes. So that's kind of one of our other motivations for this project. And so we know that these systems use different types of media to promote bacterial growth. And so there's different substrates that we've tested to see what would help grow the biofilms, what would be best for our application. We found that these bioballs over on the left had promising results. And so these are a plastic material. They're engineered to have a high surface area. So that just means that there's a lot of space and a lot of room on them for the bacteria to form their biofilm. But for our application, if we wanted to take this up to a larger scale, you would need to produce so many bioballs. And we just wanted to think of something that we could use as an alternative instead. And that's kind of how we stumbled upon bottle caps. So why bottle caps? Well, they're made of high density polyethylene and polypropylene, which is similar to the commercially available media like bioballs. And so these are also made out of high density polyethylene. We know that bottle caps are food grade plastic, so they're BPA free, and they're also readily available globally. And I think this is a really important aspect of the project because in the long run, we'd like to develop a system that underrepresented and underserved communities would be able to afford. And if you go to these communities, there's a lot of pollution problems. You probably have bottle caps, and so they're free, they're available globally, compared to our commercially available products that do cost money, and it increases your overall plastic production. Now, looking at specific surface area for bottle caps and comparing them to these engineered commercially available products, we have a surface area of about 400 meters squared per cubic meter compared to these products, which have a specific surface area up to 500 meters squared per cubic meter. And so we're really not that far off when it comes to space and space available for the bacteria to grow on. And so with our laboratory experiments, we have a continuous 24-hour flow-through system that allows us to constantly supply water that has nitrogen in it, and we can continuously monitor the processes taking place. And so every day we grab water from the lagoon and we pump it into our system. You can see the pumps in the background. We use these to control our flow rate. We can control our residence time. And so residence time is just how long the water is going to sit in contact with these bottle caps that have this developed biofilm on them. And so we're able to control these in our treatment cells. And so typically we can have 8 hours in one cell, 16 hours in another cell, and 24 hours in the outflow cell. And this, we can change this depending on the results that we're seeing in the lab. And so something that's really important to mention is that we are not purchasing this bacteria. We're not cultivating it in the lab. All we are doing is taking water from the lagoon, taking what's already existing, what is already naturally present in the lagoon, and we are just providing it with the right environmental conditions. So the conditions that it should have in a healthy and thriving environment. We are controlling and monitoring the dissolved oxygen concentrations. We're providing them with a carbon source, and we're also providing them with a home, which are the bottle caps. And so, again, a really big portion of this project is that we're working to understand these natural processes because we want to see these processes happening again in the lagoon. And this is just a picture of what it looks like when the biofilms start to form on the bottle caps. It gets this really slimy outer layer. And so scaling up this system, we do have a 5,000 gallon treatment system in place. And so this is much bigger than our laboratory system. It can treat roughly 15,000 to 30,000 gallons of water a day, which is like the size of a small in-ground swimming pool. And so it's a lot more water that we're able to treat. Now, getting to the question that I'm sure a lot of people have is this girl absolutely nuts? What about plastics? What about plastics in the environment? What about microplastics? And so I assure you this is something that we are very aware of, and it's something we're strictly monitoring. And we know that the main variables that contribute to secondary microplastics, so those very small pieces of plastic that sometimes you can't even see with the naked eye, we know that the main variables contributing to this is ultraviolet radiation, so your sunlight, and physical forces like waves and wind. Well, looking at this picture of our system, you actually can't see the bottle caps that are in there, and it is full of bottle caps, and that's because they are submerged completely under the surface, and they're also protected with a shading media, and so that's preventing any sunlight from beating down on the caps and potentially breaking them down. This is also a very low energy system, and so water is slowly flowing through it, and you don't have that aggressive wave and wind energy acting on the caps that could potentially break them down. So we're feeling confident that we're not contributing to secondary microplastics. To add to this confidence, we have been following NOAA's standard microplastic count methods and monitoring the microplastics coming in and monitoring what is going out after it's been through our system. And I'll talk about these results a little later on. But really, I just want to thank everyone who has donated to the cause so far. Your caps are out. This is just one of the many boat trips that we've taken to our site. We are giving your caps a second life. They now are happy home to our bacteria. And so thank you so much for this. And coming up, I'm going to show you some of the results. We've seen very similar results in our lab and out in the field, which shows us that this is able to be scaled up and that we can scale it up even more. I'm going to walk you through these results. And so first, looking at total dissolved nitrogen. And so I've been talking about the different forms of nitrogen a lot, and this is taking into consideration all of them. And so you can see that the system works really well over different types of influent material. And so really the main picture is to follow along with this green influent material. Influent, that is just the water that we're pumping into the lagoon. The treatment 3 is our outflow, so the water that's leaving our system, and that dashed line is representing the 3 milligram per liter, and this is a common nutrient treatment threshold. And so we've successfully been able to treat a wide range of nitrogen concentrations, which has allowed us to treat different types of water, like storm water, lagoon water, or something like dredge slurry. So even with something as high as 25 milligrams per liter, we were able to decrease it below the nutrient threshold of 3 milligrams per liter. You can see that it does take a little bit more time, so we would have to increase the residence time for something like this. And overall, we have a greater, around 70% decrease in total dissolved nitrogen. And so this has been really exciting that we know we can use this application to treat different types of water. Now going and looking at something more specific like ammonium. So I had mentioned before that ammonium is that main form of nitrogen that gets released from these muck deposits. And it's also the form of nitrogen that is favored by some harmful algae like Oriumbra. And so again, you can see that despite our variable influent concentration, so whether it's high or really low, this system works phenomenally with treating ammonium. And so we're seeing greater than 95% decrease in ammonium concentration in as little as 8 hours, very consistently. And so these are results that we're really excited about. We know that it's going to work good for treating something like dredge slurry. And so we're knocking these levels out of the park. Now we can also look at other things like molar ratios. And so if you're looking at something like nitrogen to phosphorus, your N/P ratio is usually around 16:1. And so all big pictures, what you're focusing on is that if you're seeing a decrease in your N/P ratios, that is telling you that you are preferentially removing nitrogen over phosphorus in your system. And that's our ultimate goal. That's what we're aiming for. And so we are seeing that our influent material in the green compared to our effluent material in the yellow, we are seeing ratios lower than 16, which is telling us that rather than just growing bacteria and producing biomass, we're actually removing nitrogen from the system. Now, taking a closer look at nitrogen speciation. And so just for context, when you're looking at this graph, the average influent graph is overall bigger because we're starting with an overall higher concentration of total dissolved nitrogen. And looking at the average effluent graph, which is overall smaller, that's because in the end, we have a smaller concentration of total dissolved nitrogen. So looking at something like typical lagoon water and the different forms of nitrogen that are in it, you can see that it's predominantly organic nitrogen. You have ammonium in there, and then you have very, very little nitrate and nitrite. And after these complex biogeochemical processes take place, you can see that we've virtually removed all of the ammonium, and we're left with mostly organic nitrogen, but again, in an overall smaller concentration. Now, if we change it up and we look at something like dredge slurry, you're going to notice a difference. And like I've mentioned before with the muck, it is predominantly ammonium. So we're seeing mostly ammonium in this water with a small fraction of nitrate and nitrite and a less organic nitrogen. And again, after these processes take place, we are once again removing virtually all of the ammonium. We have not accumulated nitrate and nitrites, and we're left with mostly organic nitrogen, but an overall smaller concentration, as we are seeing an average of 70% decrease in total dissolved nitrogen. So I said we were monitoring microplastics, and we were feeling confident that we weren't contributing to microplastics production, but what we were really surprised to see is that our system is actually removing microplastics. And to date, this is a significant difference from what is coming in versus what is coming out. And so we were really surprised, but really excited to see these results from our system. And I think it can have a lot of potential benefits in the future. Another associated benefit that we weren't really expecting was improved water clarity. And so although water clarity doesn't necessarily mean water quality, this is really beneficial for those submerged aquatic vegetation like seagrasses that require sunlight. And so with clear water, sunlight can penetrate deeper and they're able to carry out photosynthesis and produce oxygen. And so this is our untreated water versus our treated water. And there's a significant difference between our influent turbidity levels and our effluent turbidity levels. And so we're actually lowering the turbidity with our system as well. And if we take a step back and think about plastics and why they're a problem in the environment in the first place, we know that organisms can get entangled in larger plastic pieces. We know that they can be ingested, but we also know that plastics sorb harmful contaminants. And so in this application, if we have these bottle caps completely confined in a system and we're able to have control over them, we've been interested in seeing if we're actually able to remove these contaminants as well. And so we are currently waiting for results of samples that we had sent off to a lab at Florida Tech to evaluate if our system is actually able to remove organic contaminants. And I'm really excited to see these results, and hopefully in the future I can give another update talk. Another great part about this project has been its outreach opportunities. So getting to do presentations like today, where I get to talk with the community, hear your ideas, answer your questions. I've talked to many elementary schools, middle schools, and high schools, and this has just been a great opportunity to raise awareness about plastic consumption and plastics in the environment. And so you can kind of think, if you're collecting caps, I'd like you to think of them as like your own personal token. And so this is how you can kind of keep track of your own personal use. And I hope that your goal is to make your pile and your contribution smaller and smaller as you find alternative uses. It's also been a great opportunity to let people know about the Indian River Lagoon and also environmental research taking place at Florida Tech. And I really just like to encourage everyone that everyone has the potential to make a positive change and a positive impact. This project has really showed me that the community really wants to help, and you guys want to be involved. And as scientists, we really appreciate this. And I think it's really important to remember that you as an individual have the potential to make a positive change, and that you can make this change in your community as well. And eventually, if we all come together and keep working at this, hopefully we can help restore the lagoon. So this is just some of the schools that have been on board with this project that I've either presented to, or they've been collecting caps, or they've reached out. And so lots of schools in Brevard County. So thank you to all the teachers and students involved. And some of the key takeaway points is that I'm sure some of you are concerned, maybe initially, we are not just throwing caps directly in the lagoon. As you saw, we have built a treatment system. We will hopefully scale this up in the future, but regardless of the scale, these caps are completely contained. It's not leading to the production of secondary microplastics. This has been a great opportunity to raise awareness about plastic in the environment and plastic consumption through educating local schools, organizations, businesses, and the community, encouraging people to reduce their plastic use, and also raising awareness on marine debris. And so it's always really exciting when people come in and they donate bags of caps that they've actually collected from the beach. And they say that they would have otherwise not noticed the caps there. And I'm sure after this, you might start to see them more. I hope you pick them up, remove the trash from the environment. And as for the project update, I cannot thank you enough. We are just so excited by the support that we have received in this project, and we were never expecting it to take off like this. And we're happy to say that we've already reached our quota for cap collection. And so with this being said, we are pausing our cap collection until further notice. And this is your support and the immense support has allowed us to be more selective and look for solely bottle caps. And so before you're too discouraged, if you already have caps that you've been collecting, we will still happily take them. We will happily take your bottle caps, and the main drop-off locations for these are still located at the Sunshop Cafe in downtown Melbourne, Brevard Zoo, and the Tiny Turtle in Cocoa Beach. There is also an additional location at the Dixie Crossroads Seafood Restaurant near Titusville, so a shout out to Chelsea for getting all of that organized up there. And I've also received some emails regarding potential volunteering opportunities. So I know MRC sent out a social media blast that we were looking for volunteers, and this is true. Right now, we are still working out the logistics from the university. We do have limited space, so we were looking for a very small amount of volunteers in the future to help us sort through the caps that we've collected. For those, if you've emailed me and I haven't gotten back to you yet, or if you're interested for the future, I will reach out to you when this time comes when we have it figured out. In the meantime, though, it sounds like with MRC, there's plenty of volunteering opportunities to go around, and there's always volunteering open in the community, so make sure you keep your eyes out and see other ways that you can help. Ultimately, with this project, we're continuing to understand these processes within the environment, and so it's really important. It's cool that we're able to make this treatment system, but unfortunately, this treatment system shouldn't be necessary in the first place. All of these processes are naturally occurring within the environment. And so this is really motivation for us to strive and to continue to understand these processes so we can promote them in the lagoon, and we can promote these healthy sediments and healthy lagoon overall, so these processes can be carried out and we can see natural nitrogen removal like we're supposed to. We can use our laboratory experiments to kind of further understand these processes, to take what we've read from the literature and apply it. And then we can take what we've learned from these experiments in the lab and go out and apply it into the lagoon. I think it's really important to mention that all of the work going towards the lagoon, this is all a collaborative effort. It's a multifaceted approach, and it also takes time. And so it might be frustrating to not see the results right away, but all of these processes take place. It takes time to restore these ecosystems, and it takes time to promote these ecosystem services. But with the community support and everyone's support, I think we're well on our way. For our long-term goals, we would like to continue to scale up the system and treat even more lagoon water. And so we are interested, we have talked with municipalities, we are very interested in scaling up this system. And so on the other hand, we'd also kind of like to scale it down a little bit and use something like a rain barrel to create smaller, more affordable treatment systems. And so thank you to Marine Resource Council for donating our first rain barrel. Our goal is to hopefully have one of these created so that in the future, it can be used potentially by residents. And so although it'll make a small difference, it's not going to magically fix your lagoon. Like I said, it's a collaborative effort for restoration, but it still will make a difference, and it's still going to help promote these ecosystem services. We would also really like to get these up and running, these smaller treatment systems, again, to get them implemented in the underrepresented, underserved communities that might not be able to afford other treatment systems. But our main goal is that we're using this research, we're using everything that we're learning in the lab to help apply this to the lagoon. So I know Marine Resource Council recently released their lagoon report card, and one of the pieces of this report card is water quality. And so the variables within this is nitrogen, phosphorus, turbidity, dissolved oxygen, harmful algae blooms. And so really everything about this system and all the research that we've been doing in our lab is kind of tying into all of these and helping us learn what we need to do to promote these ecosystem services in the lagoon. And so we have been able to achieve so much with this project in a pretty short amount of time. And really a big part of it is with the community and businesses stepping up to the plate. Organizations. Thank you to Brevard Zoo. They've been great with having donations. All the schools that have gotten involved, it's been so fun to watch all the students get involved and to talk to younger generations and encourage them because they're our future. And we have gotten a boatload of caps. And so you saw the picture earlier with me taking them over to our big system. Your caps are going to good use. They're now home for these great bacteria. And we're really excited about all of this. And so again, this has been a huge collaborative approach. I really need to give a big shout out to our lab tech, Stacy Fox, because she helps with all of this nutrient analysis. And we are go, go, go with this system. We are constantly making modifications. We're constantly learning about them. And so she has been a huge help with this. And a big thank you to my advisor, Dr. Austin Fox. He's always sharing his passion for helping the lagoon, and his patience and his guidance along this research has been absolutely great. And thank you to Tyler. He works over at Brevard Zoo. He's been absolutely great coordinating us with them. And also thank you to Anna for your support with this project and help in the lab. So really, thank you to everyone. Everyone in this community has been a part of this project. We're really excited to keep it going. If you have any questions, you can reach out to me. You can also follow our Facebook page. So we really like to see people posting on our page. If you're collecting caps, if you've collected caps, we like to read emails. I'll leave it off on this note. One of my favorite responses so far has been from a father who his 7-year-old was collecting caps for his school. And he had asked him, what is the prize for collecting the most caps? What do you get out of this project? And his son responded, well, Dad, the prize is a clean river. And so those are the things that really keep us going. At least it keeps me going. That stuff is absolutely heartwarming, and I'm really glad with this impact that we're having on the community. And so with that, I will be happy to answer questions.
Awesome job, Abby. Thank you so, so much. We've gotten so many questions about this project and how to get involved and where to drop off caps and what kind of caps. And, up until now, I've been like, I'm not sure. I'll find out. We can find out. So when your name came up as a lunch and learn presenter, we were like, that's brilliant. Like, we need to have her come speak to us. So thank you so, so much. We have gotten a few questions in, and I'm just going to reiterate because a few of them have been about drop-offs. And correct me if I'm wrong, you're pausing collection at this time?
Yes.
Okay.
We have plenty of bottle caps, and so once we get the logistics and we find a couple of volunteers, we would definitely appreciate some help sorting through these caps that we've collected.
Perfect. And this email here on the screen is the best way to get in touch with you about volunteering, correct?
Yes.
Okay. And if you do already have caps, there are, again, a few locations to drop them off at Sunshop Cafe, downtown Melbourne, Brevard Zoo in Vieira, Tiny Turtle in Cocoa, and Dixie Crossroads up in Titusville. So in case anybody missed that. All right, let's see. One question here from Ruth. How long were the bottle caps in the water before you started to see some results?
And so that's a great question. It definitely, so you'll see a lot with these systems is that it does take time for the microbial communities to establish. So one of the really cool things is when you take a step back and we can look at the results from the beginning, you can actually see the improvement of the efficiency of the system. And so it takes roughly, it didn't actually take long. So in the lab, maybe I would say like a rough estimate of 10 to 14 days. But almost right off the bat, you do see nutrient removal, but it definitely, as you start to see the biofilms get thicker and you actually see those caps coated with the films, that's when we see these higher efficiencies.
Okay, interesting. Another question here that I thought was very interesting. Have you done any experiments to manipulate the structure of the caps and the removal rates? Like, for example, bottle caps, does air get trapped underneath it to slow down the mixing motion? Does efficacy of denitrification decrease over time as detritus builds up on the biofilter? Very, very interesting question.
Yeah, that's a great question. And so with the efficiency, decreasing efficiency, we have not seen that so far, but we have been working with the system for quite a while. And so I think that as long as obviously, if you were to scale it up, you would have to have some precautions, to prevent biosolids from forming. But when you look at those N to P ratios, you can actually see that it's not just the biomass that's removing the nitrogen, it's the processes that are removing it over phosphorus. And then, can you reiterate the first part of the question?
Yes. Have you done any experiments with manipulating the structure of the caps and to affect removal rates? For example, air getting trapped underneath the caps.
And so we actually found with the bottle caps that they did work really well because they do kind of create their own environment of oxic and anoxic conditions. And so with air getting trapped in the caps and then with the biofilms on the caps, it's actually ended up creating this ideal condition for the bacteria. And so I showed that picture earlier on in the presentation of all the different media. And that's kind of what we tested before. And I guess the bottle caps can be seen as like a natural manipulation of what bioballs are, because it's the same material, but it's repurposed. And so we were surprised that they had worked as well as they have been working. And so I think they've found their successful repurposed life.
Yes, for sure. A question on the longevity of the project and where you see this going. Like, what applications do you hope to use this in, like on a greater scale, lagoon wide or even, globally?
So ideally, I mean, it's always the goal if we could get this implemented globally, especially because, we're really trying to get this developed inexpensively because we want it to be something that communities that otherwise might not be able to afford treatment systems, they can afford it. And so that is ultimately the greatest goal that we could have. And we are always looking to expand this system. And so because I showed you those various influent nitrogen concentrations, we've proven that this treatment system can actually treat different types of water. And so we would like to work to treat lagoon water or storm water or something like dredge slurry in the future. And so because it has such promising results removing ammonium, it could be potentially used to treat something like dredge slurry.
Yeah, or even in like a baffle box type
Exactly. So that was my next thing, is that we would be interested to see if these can get implemented in baffle boxes to treat stormwater, as long as they're completely submerged, because I know some baffle boxes are dried out.
Their water level can vary. Yeah. That was the first thing that jumped to my mind. I'm like, this would be a fantastic addition to baffle boxes.
Yeah, and so with every project, you hit bumps in the road, some things that take you back, but we're still moving forward and we are working to get this developed on a larger scale. We've talked to municipalities. We're still looking for interest in sizing this up even more.
Awesome. So if anyone has any contacts, feel free to email Abby at the email on the screen. All right, I think we've got time for just a few more. After the bottle caps have done their job, what do you do with them?
And so we've actually been able to continuously, because we haven't seen breakdown of the caps and because this hasn't been applied at the largest of scales, we haven't run into that problem yet, but we've already started looking into alternatives. And so there are organizations, like I know it was common around here with the Lions Club, they collect caps and they send them off to make buddy benches. And so we would like to, obviously not, like we've done this much with them, and so we would like to give them even another life after this use. But ideally, idea. Because we're not seeing the breakdown of the caps, they're very durable.
Great. That's a great idea. All right, let's see, one last one. Okay, the nitrogen reactions require energy that bacteria typically get from reduced carbon. Do you add a carbon source to your microcosms to support their metabolism?
Yeah, we do add a carbon source, and we're also testing different carbon sources. That's one thing that's part of my thesis, is looking at how different carbon sources can impact it.
Awesome, awesome. Everybody, fantastic questions today. Thank you so, so much. You are a very engaging audience. And Abby, awesome job on your presentation. Congratulations on such amazing work. We need this and so much more going on in the lagoon. So thank you for starting a really
Thank you.
fantastic and engaging master's thesis. We're lucky to be able to have you come and speak to us today, and we're excited to see your next steps and how you apply this farther down the road and, you know, on a larger scale.
Well, thank you, and thank you everyone for listening and joining along.
And again,
I love talking
everybody,
about this.
if you have any questions, feel free to send her an email with her contact on the screen. We'll also send our follow-up email will include her email address so that you can reach her in case you miss it. And Abby, thank you again, everybody. Thank you for logging on. Our August Lunch and Learn is going to be on low impact development with Joni Reagan formerly with the city of Cocoa Beach. So you'll start seeing email signups for that. So keep an eye on your inbox, and everyone have a fantastic rest of your Tuesday. Bye, everybody.